She said we saved the best for last.
Okay, good afternoon, everyone. This is going to be the last talk, so bear with me for the next 15 minutes or so. By this time, I have to say that each and every one of us has probably appreciated how devastating Alzheimer's disease is. And from the past talks, you have seen that amyloid beta deposit is actually one of the hallmarks of Alzheimer's disease. So today I'm going to be talking about some very exciting research in my laboratory towards developing some therapies for Alzheimer's disease. So essentially, the therapy that I'm going to be talking about is based on a physiological pathway, which is very, very conserved in the cell, and that is based on a process called phagocytosis. Essentially, when you talk about phagocytosis, it just literally means cell eating. So it is the process of eating dead cells, cellular debris. It is also the process that's involved in clearing out pathogens. It's very, very important in immune response, as you could imagine, for example, that the autoreactive T cells and B cells need to be cleared. It is very important in morphogenesis because as it happens, we do need during retinogenesis, for example, as well as development, we need to clear those neurons that are actually immature. And during tissue damage as well as repair, and in the case of disease conditions, for example, as in age-related macular degeneration, wherein drusen need to be cleared through phagocytosis. Phagocytosis could be classified into the two general pathways. One that elicits an immune response, and that particular pathway actually utilizes a specific receptor that is the receptor for Ages. And that particular pathway results to inflammation. The other pathway that I'm more interested about is what we call as the silent pathway. For example, in our eye, every single day, the tips of the photoreceptors are actually shed. We don't notice it. Phagocytosis is going on because it is very, very silent. And that particular process is actually mediated by another receptor called tyrosine kinase. Okay, so where does our phagocytosis-based therapy play on this? And what do we know about the clearance of amyloid beta? Essentially, in the brain, the major cell that's responsible for the clearance of amyloid beta is actually the microglia. And when microglia clears the amyloid beta, it goes to this inflammatory cascade in such a way that it releases all these inflammatory cytokines that eventually results to death of the neurons or the brain cells. This is not what we want to happen. So our goal in the laboratory is actually how to divert the clearance of amyloid beta into another process, which is through the process utilizing the silent receptor tyrosine kinase. Okay, but essentially on this pathway, what happens is that the clearance of amyloid beta does not result into inflammatory response. So essentially, the cells are still going to be healthy. So how do we do that? So the idea that we have is actually to sequester the amyloid beta and target it to the MER tyrosine kinase by creating a hybrid molecule that could bind the amyloid beta and target it to the MER tyrosine kinase pathway, which is the silent pathway. So from my previous studies, I first worked in the eye, and I utilized MER tyrosine kinase looking at phagocytosis in the eye, which is a silent pathway. What I found was a specific protein, TABI protein, that is actually acting as a ligand for phagocytosis. So it is a signal that links up the photoreceptor outer segments into the MER tyrosine kinase. So here is the protein. What we know is that it has a signal that recognizes the MER tyrosine kinase, and the C-terminal domain of the protein is the one that is responsible for linking up into the photoreceptor outer segments. So the process in the eye happens such that TABI sequesters the outer segments and of course targeted into the MER tyrosine kinase all the way into the phagosome, and yet you don't produce any inflammation. In the alternative pathway that we are trying to do, our hybrid protein would be able to get amyloid beta and target it into the MER tyrosine kinase pathway for liquidation. So the strategy that we did actually involved 3 stages. It's a combination of bioinformatics in such a way that we lined up all known amyloid beta binding proteins and then looked for a possible consensus sequence. The next stage that we did, once we found the consensus sequence, is mutate the sequence or evolve the sequence in such a way that we could find an amyloid beta binding peptide that should be able to bind really, really highly to amyloid beta, so that it could act as a competitor against the natural receptor RAGE. After evolving the sequences and mutating the sequences, we put it into a T7 phage vector so that we could create a library of phages containing different mutants with amyloid beta binding proteins. After that, we did the screening and looked for one that actually bind to amyloid beta. So we tested both the amyloid beta fibril as well as amyloid beta oligomer. As we know that in the plaques, it's mostly the fibril form that is present. However, the most toxic form is actually the oligomer. I want to focus your attention to peptide number 6 right here. So what we found is that peptide number 6 is actually very, very specific against the oligomeric form. So knowing that this is the most toxic form, we know that we probably could use it later on for therapeutics. So we pursued peptide number 6. So the strategy that we did, now we have peptide number 6, we'll link it out into the N-terminal of TABI. So we made 3 modules of peptide number 6, and also we made 3 modules of the TABI N-terminal containing the domain that specifically binds to the MER tyrosine kinase. The idea is that the hybrid protein should be able to sequester amyloid beta, targeted again to MER tyrosine kinase pathway without producing any inflammation. So, but first, we have to see whether when we have the hybrid protein, does it even bind or does it even recognize the receptor, MER tyrosine kinase? So in this case, we look at the binding of our hybrid peptide to the MER tyrosine kinase receptor. So here, as you can see, this is hybrid protein number 6. And we compare it to the ability of TABI protein, which is the parental form. And the binding is higher, but not significantly different to that of TABI. bind to amyloid beta oligomers. So we tested its binding to the oligomers, and we found that hybrid number 6 actually significantly bind to amyloid beta oligomers. In comparison to the natural receptor range, as shown here, it is almost 4 times higher than the natural receptor range. So this gives us an idea that we could have, or we were able to generate a good competitor against the possible natural receptor, and something that could act as a decoy and sequester amyloid beta to the Merck tyrosine kinase pathway. The next question that we ask is, this is all happening. Those that I showed you are all happening in vitro. So what does happen in vivo or within the cell? Can the cell take up amyloid beta? Or in the presence of the protein, can it actually facilitate uptake of amyloid beta? So here we tested whether hybrid number 6 could facilitate the uptake of amyloid beta. MVP only is our control because our protein is conjugated to a multiple spike protein. So we did a phagocytosis assay on microglial cells, and what we found was that yes, it is able to facilitate uptake of amyloid beta. And compared to the control, it is significantly different compared to just MVP. The next question that we asked was, how specific is the phagocytosis or the uptake of amyloid beta as facilitated by our hybrid protein? So we did some blocking of the receptor by adding a MER-FC, which contains the extracellular portion of the Merck tyrosine kinase receptor. And what we found was that there was a significant reduction in the uptake of the production of oxidative products. So here we look at the production of nitric oxide. And what we found was there is a significant reduction in the production of nitric oxide. And that reduction actually follows a dose-dependent manner with a very significant decrease at a level of 50 nanomolar to 200 nanomolar. And the same trend is shown in 24 hours as well as 48 hours. So in our hypothesis, we said that if we divert the clearance of amyloid beta into the Sited pathway or the Merck tyrosine kinase pathway, there should be a reduction in inflammatory response. So we'll look at some of inflammatory cytokines, as well as some anti-inflammatory cytokines. So in this case, we look at the level of TNF-alpha, IL-1 beta, IL-6, as well as IL-10. So TNF-alpha and IL-1 beta are very, very inflammatory. And as you can see, there is a reduction in the level of TNF-alpha when you look at hybrid number 6 in blue right here. For IL-1 beta, there is also a significant reduction. However, when you look at IL-6, there is no reduction. The level is almost the same in both the treated with hybrid peptide as well as in control. However, when you look at IL-10, there is a significant increase in the level of IL-10 when it was treated with hybrid number 6. IL-10 is anti-inflammatory. So from this result, we know that somehow we're getting some reduction in the level of inflammatory cytokines and an increase in the level of anti-inflammatory cytokines. But I have to say that this data is very, very preliminary. There's going to be a lot of work in really looking at whether this is a very consistent result or not. Okay, so we know that we have peptide number 6 that could potentially be used for therapeutics. But it's always a problem. If you have a potential drug, can it even go to the site where you want it to go? In this case, this is the brain. So we tested whether peptide number 6 actually could cross the blood-brain barrier. And we did that by injecting a wild-type mouse with hybrid map number 6, and the other one, which is the control PDS. And we sacrificed the mice after 30 minutes, and another set was sacrificed after 1 hour. And we look at whether hybrid number 6 using antibody against the tag to our hybrid number 6 after 30 minutes and 1 hour. So what we found that after 30 minutes, hybrid number 6 is actually found in the hippocampus of the mice. And there is a significant increase of those that were able to cross the blood-brain barrier after 1 hour. So this is just a control secondary antibody to show that the antibody we're using is actually quite specific. In contrast, in a control injected, you don't see any reactivity against the antibody. So just to give you a summary, we have generated a new amyloid beta binding peptide that binds to the different forms of amyloid beta. We don't have some that binds to the fibrils, but for now, we are actually pursuing the ones that binds against the oligomer. We also have engineered the hybrid protein, and we have shown that that hybrid protein reduces oxidative products as well as inflammatory factors in the microglia. And we have shown that the hybrid protein can actually cross the blood-brain barrier. So where do we go from here? Essentially, we only scratch the surface. Okay. So essentially, we need to do some mechanistic studies in the future and really look at whether there's an effect for amyloid clearance of amyloid beta. And with that, I just want to thank